Our next speaker is David LaPuma. Hi, David. David is Vice President of Cellular Tracking Technologies, an environmental tech company in Cape May, New Jersey, and he's going to be talking to us about how new technology is linking community science and banding station research. Thanks, David.
Thank you. First, does anybody get the reference? Thank you. At least a few people got it. I was at the bar last night with one person who was younger than me and older than me, and neither of them got it. And I'm like, oh no, I picked the wrong song. Well, if you don't know, go ahead and YouTube it. You can listen to it. It's a great song. But anyway, I'm going to talk a little bit about frequencies. We've been talking to every speaker today has kind of mentioned the MODIS network in some fashion. And the frequencies I'm going to talk about are the MODIS frequencies and some new frequencies that are coming online. But really what I want to talk about is how we can use this new technology, this emergent technology, to connect more people to the research we're doing, leverage more conservation literacy, but also leverage some support. So who here has to hustle funds for their banding research every year? It's okay, hustle fund. Who here has to raise money to do their work? Raise your hand high. There's only a few of us in the room. Right on. Great. Yeah, a few of us do. And a lot of the stuff kind of came out of when I was the director of the Cape May Bird Observatory, every year having to raise the money for the hawk watchers, for the sea watchers, for the banders, the new banding station. It's an important thing that we have to do. And the more we can leverage some of the work we're actually doing to engage people, I think the more effective that can be. So let's see. Am I going to figure out how to do this?
David, could you use the mic, please?
Let me see if I just turn this on, will it work?
Okay.
Ground control to have. Got you. All right.
Okay.
Let me see if this works. I still don't know how to forward the slides here. Caitlin, how should do that work? Spacebar? Awesome.
Oh, I didn't even know that. No, so I literally just put my cursor on the arrow, put the cursor there, then I just kept clicking it. Yeah.
Cool. Can do. All right. Small little agenda. We'll talk a little bit about how we bring our data to the masses, some wildlife telemetry today. So what are the new technologies that are available now? And then this blue revolution, which I'll talk about. You can also see at our booth out in the hall. Oh, now the arrow works. Okay, so first bringing the data to the masses. Again, this goes back to when I was at the Cape May Bird Observatory, where we were still collecting data on paper and then putting into Excel, and nobody saw it but the folks that were at the Bird Observatory. Not only that, we had to actually hire people to check that data at the end of the season and re-import it or import it from the paper data, which was laborious, expensive, and again, it's in the dark. So I know some of you are already doing this, but I did give this talk at the International Bird Observatory Conference, and it was very pertinent there because there are resources out there where you can put your data in digitally immediately, so you don't have to transcribe it later. It goes into the cloud, it's managed for you, and you can display it online in ways that you can engage donors. And we did this at the Bird Observatory, and it's been super effective in getting people excited. People can go on to the Trek Tellen sites. It's a Dutch site that has thousands of sites now where people upload their data. And they can look at the different counts in real time. So, and this is a banding, a ringing site, because it's Dutch. So, but it's a banding site. But then also we've got all the visible migration counts as well. So I'll give you kind of an example of what that looks like. So here's Friday the 13th, October 2023. A really nice count of the hawk watch. Something you'll also notice here is it's not just hawks. This digital data entry allows us to enter everything from all the species of birds, but also common buckeye, monarch, red admiral, so red bat. So we're putting in all the data. So we're actually capturing a lot more of what's really going on in this visible migration in a way that's easier to do than it ever was on paper data. And it's available to anybody who wants to look at it. These are all banding stations. And I'm only looking at the Americas. So this is the true Americas, right? So Canada all the way down into Mexico. And you can see, and in the Caribbean as well, we got Havana banding stations. So we go back to our station on that same day, Friday the 13th, October 2023. So how many times do we talk about what is our visible migration count data mean in terms of our banding data or vice versa? Now we can look at those things side by side and see that. So this is just the banding data. You saw the visible migration data. Again, a very powerful tool for connecting people to the work that you're doing at your station. Obviously, there's been a lot of interest in the MODIS network, and this kind of eye candy here gets people very excited when you can see all the birds that are moving through the network. So these are animals tagged and detected in South America, summer to fall 2023. That is powerful imagery to engage folks that you're trying to get both to understand the work, the importance of the work you're doing, and also eventually to support it. So at our banding station in Cape May, this is a partnership between the Nature Conservancy, which has the property. This is the South Cape May Meadows. If everybody here has gone birding in Cape May, should have if you haven't. New Jersey Audubon, where I was the director of the Bird Observatory at the time, and Cellular Tracking Technologies, the company that we were partnering with and who I work for now. That's our MODA station above the information shed at the Meadows. And here's just some of the data. So 2023, we put a lot of tags out. And so we've got northern water thrush, veery, red-eyed vireo, and swamp sparrow. These are all the ones that were detected throughout the network. So a lot of cool movement data. Again, this type of data going back to the funder who funded the station is super, super powerful. What I'm not going to show you is we've actually got some node data at the site where we can see habitat use of these birds, which very locally is very effective for engaging the Nature Conservancy, who wants to know whether the restoration work is effective. And they can test that directly with empirical data on the site. But I can't talk about everything today. So I want to talk about some of this stuff. So wildlife telemetry today. I don't know about you, but I've been doing this for a good decade in terms of helping people get transmitters out. And only 2019, we were trying to get a transmitter that a GSM transmitter that can send data over the cell network down to 30 grams. Now we're at less than 3 grams to send data over cell network. So the changes have happened super rapidly in very short order. And now we're talking about transmitters that are weighing the same as a grain of rice, or 60 milligrams, 0.06 grams. Talk about that one momentarily. But so some of you study small raptors. Here's a sharp-shinned hawk. Up until very recently, sharp-shinned hawks, the only thing you could put on them was a nano tag or a life tag or a hybrid tag, something that weighed, sub-gram, or the only thing that was small enough was a radio tag, I should say. They can take 3 grams. A male sharp-shinned hawk could take a 3-gram transmitter. Female could take a little heavier, but it just didn't exist. Well, now we have transmitters that send data over the cell network. They do a couple things. One, they treat the cell network like the MODIS network. That's what these blue dots are. These are just cell locate pings. And so they're pinging, they're turning on, they're collecting data about the cell network around them. They're sending that data to a server, and the server is going, okay, I think you're here, plus or minus 300 meters to several kilometers, which if you're used to Argos, is very similar to an Argos Doppler fix, except that's a lot more data than you might get on with an Argos device. They also have a GPS on board. So it's a small transmitter, it's got a small battery, so you got to be very mindful of how much power they're using, right? So we can leverage that by saying, okay, we just want to do some of these cell locates, and then every once in a while do a GPS fix. And at this level, at this landscape scale, or this continental scale, the two line up very well and tell a similar story. So this data, which is very easy to collect, doesn't take a lot of power, actually tells us a lot about that broad scale movement. And we can get some specific locations. We want to know where the bird roosted, right? So kestrels are another species that until very recently we didn't have a small enough tag that could send data besides hitting a MODIS tower necessarily. So again, same thing, cell locate things here and GPS things here, and then some GPS at a finer scale. So we can really look at wintering habitat use of these birds that we could never put some kind of, the only thing you could put on one of these birds was a logger that you had to recover later. So we're talking about like juvenile dispersal, right? We want to know where these birds go, but they're not going to come back to a box necessarily. This allows us to do that by remotely sending that data over the cell network. And I referenced the sources for these data as well. This is those folks out of Manitoba who are doing saw-whet owls as well. Speaking of saw-whet owls, a couple of years ago, we put our first cell locate. So this one only pinged cell towers at the time. Now they do GPS. We put it on a saw-whet owl in Cape May. We were testing out harnessing. We put 6 out, 5 of them with stretch magic, 1 of them with a spectra material that we found. And this is the spectral one. So the ones with stretch magic, we think, bit through that and got out of it. I think the weight combined with the weak material allowed them to get away, get out of it. But this one individual gave us so much data on both the overwintering. So this bird didn't leave Cape May until April 25th. So we were like, we knew it was alive because we had activity data from the ACC on board, and every day it would check in. It would send us activity data, so we knew it was active. But it wasn't really moving. It was just pinging the same cell tower. And April 25th, it starts to go north, and we tracked it all the way up in its migration.
So where did it end up?
It ended up in Maine. It's up right outside of Stratton, Maine. And then this was only meant to last about 6 months. So after 6 months, the battery died. And so it was up there for a while. So we think that's where she was settling in to nest. But now we have better batteries, we have smaller units, and hopefully we can start getting some of that return migration. We can do it with solar rechargeable units, but these battery-only units are a little tougher. I don't have a clock on here. I have no idea how long I've been talking. All right. So the next step is this blue revolution. It's called the blue revolution because we're using a new frequency, 2.4 gigahertz, which is the same frequency as Bluetooth. So the beauty of well, let's back up. Right now we've talked about cellular units, we've talked about radio units in the Modus network. Those all require infrastructure. Obviously, the cell network is there. The Bluetooth units or the 2.4 gigahertz units have the opportunity to leverage some of the technology that's in your pockets. I'll talk about that soon, but here's the blue morpho on a monarch butterfly. Here's the blue morpho on a, oh my God, we were just in Mexico doing hummingbirds. I'm gonna miss that one. Here's a white-eared hummingbird. And then here's the blue morpho against some stickers so you can kind of see the size. And I didn't, unfortunately, that's Japanese yen, so it might not be easy for everyone to know about this. I got stickers. So very, very tiny, tiny unit. You can see it over on the table outside. So here's the Modus network, right? So we've talked a lot about the Modus network. Modus network is fantastic. Folks in this room have been just revolutionizing the expansion of the network. And it was great to see the talk today about this kind of gap in the Hudson Valley, because the only reason why there's a gap in the Hudson Valley is because there are stations all around the Hudson Valley, and that did not exist very long ago. So it's wonderful to see the work that's been done through the Northeast Modus collaboration. So again, how many Modus stations are there? Despite the fact that there are so many, over 2,000 stations out there, it's still a chain link fence, right? There's still ways to sneak through that, and we see that in the data. How many cell towers are there? Well, cell towers are more ubiquitous, right? They're meant to so that you can go from point A to point B and you'll hit many towers and you'll never drop that call. So we've seen from some of the cell data that we can really get some great data out of those small units. But how many cellular phones are out there, right? So every one of these devices that's in your pocket that has Bluetooth now is a potential receiver. And so how do we leverage that? And I can tell you just from the example I'm going to show you momentarily, this has really been eye-opening, and we expected it to be popular. It blew up, and I think it has the potential to affect a lot of projects in this room. So obviously, this is not the Eastern Butterfly Banding Association, but we can pretend today, or as I told Joe earlier, just forget that's a monarch and think of it as a purple mark. So there's a long-standing history of tracking monarch butterflies in Cape May. Cape May's got the longest-running monarch monitoring project, started in 1992. They've put out over 250,000 tags. They've had thousands of volunteers. The Monarch Watch. This is the one in Kansas. So over 250,000 tags, thousands of volunteers. The Monarch Monitoring Project is in 1990, so it predates the Monarch Watch by New Jersey Audubon's Cape May Bird Observatory. They do both a road census and they do sticker tagging and public demonstrations. And they have hundreds of volunteers over the years who've helped put out these tags. So here's kind of some of the data from the road census. So a lot of noise, pretty flat. The take home from that is actually that the eastern monarchs that are coming through, the butterflies that are coming through Cape May actually seem to be doing pretty okay in our area. And there's a lot of reasons for that. There's a lot of habitat created. It's basically, they've created like this way station on steroids, lots of nectar plants and things that they like. And so they can do pretty well in Cape May when they get funneled to Cape May. That's not the case for monarchs across the range. And so there's a monarch with a tag on it, an illustration of a monarch with a tag. And then these tags get resighted or refound, just like bird bands, sometimes on dead ones, especially when they get to Mexico. The recoveries are pretty much all on dead monarchs, and many of them throughout the range there. Some of them get recaught as live and released. But it's few and far between, way fewer than bird banding. And that's, we know, is also a pretty low number. So the goal here was to take this real interest in conserving monarchs and find a way that we could better track them by leveraging, again, that Bluetooth option. And the tag is not that big, it's way blown up. So we're going to utilize some of this groundbreaking technology. We created a community science project to crowdsource detections of monarchs with your phones, as I mentioned, which would increase public scientific and conservation literacy by directly engaging them in tracking research. We hypothesized this would happen. Kind of, if you build it, will they come? So we use our new tags weighing only 60 milligrams. They transmit again over this Bluetooth frequency. They don't just send their digital ID though. They also send temperature and solar voltage, which for you folks who are putting out battery tags is going to be really important because the battery version sends the battery voltage. So not only do you know whether you get a detection, you can tell where your battery's at. So when they start to blink out, you can say, well, they're blinking out because the battery's actually getting super low. And the solar ones are batteryless designs. So they can theoretically last forever. So we created an app for this, right? Because you had to have an app for it. And the goal was to allow community scientists to do some of the heavy lifting for us to collect the data, but we had to give them some incentives. So we gamified it to allow them to earn badges and get status and move up and down in kind of a leaderboard. Who found the most monarchs? Who got the most detections? I could tell you that we had folks who would find out we were doing a release and would drive an hour and a half to come be there just so they could get higher on that leaderboard. So this works. This is a dopamine receptor effect, I'm sure of it. We tagged 75 monarchs. We tracked individuals from 1 to 12 days in Cape May, collected more locations on monarchs than ever before in the history of science. And we had 3,000 downloads of the app and about 300 plus participants actually actively out there. And you can download the app now. It's Project Monarch on both app stores, and it's free. And you can kind of see some of this data. So basically what that looks like is, here's a monarch named Kashi. So they were named after people who were involved in the project and some of the eminent ecologists who worked on monarchs. I mean, these are all ways to get people excited. You've got time since detection, signal strength, distance from the release site. And then from there, you can kind of look at a map. So basically what we have here is the deployment site is the star, the blue dots are the detections, then the butterfly is the current location. So you could actively look at all the different butterflies that were tagged and where they are at the last time they were seen. So most of this occurred on Cape May Point. We didn't expand the project very far outside of Cape May, so we don't have detections beyond Cape May. That's why we only have detections up to 12 days. But this is the proof of concept, I think, that's going to take us into the full flyway this year. And so this is what it looks like if you look at all the monarchs at one time. So again, mostly on Cape May Point, some up the road, Mike Land Zones, houses right here. So you can tag one named Mikey. So again, extremely engaging, lots of support, and the future of this is to build out the app in a way that anybody who wants to could participate, deploy tags. We've already got this happening on the California monarchs, the Western monarchs, which is super exciting. So again, it allows scientists to better understand monarch migration routes, the timing of monarch migration, stopover habitat location, and duration. And then we can do, focused conservation and restoration works based on the data and hopefully inform some policy focused on monarchs. And again, I say replace monarch with purple martins because monarchs are a great example because they're in this one locale, but anything that's nesting in a fixed structure that we have access to, that especially with we saw Joe's talk last night, purple martins they require human intervention at this point to survive. So humans are very much a part of that. This is an absolute logical extension to help monitor the purple martins and look at natal dispersal. So our 2024 goal is to upgrade the Eastern Monarch Flyway with these new adapters. So basically, this is a blue receiver. This is down in Mexico. We're doing our hummingbird work about a month and a half ago. And so the blue receivers up here, these are the 2.4 gigahertz antennas. And there's a little 5-way kind of adapter to put on top of an existing motor station. So that whole thing, I can hold in one hand and put it up on top of the station. It's not like having to put up giant Yagi antennas to add the blue receiver. And if you put up a motor station before, you probably dealt with lots of coax, which is both expensive and unwieldy. This is 3 feet of coax basically connected to the receiver. So we're trying to make this as simple as possible so people can just go out and either bolt it onto an existing deployment or take a piece of conduit, stick it on top of it, and just run the cable to the station. We want to make it super easy so we can get as many of these out there because I banned ruby-throated hummingbirds. I want to put these on ruby-throated hummingbirds. Many people want to put these on monarchs and other species. So we got to get the station or the stations upgraded. So in conclusion, emerging technology can increase opportunities to engage new participants in your conservation work. Leveraging that community science helps increase the impact you can have at your banding station. You're teaching people about the research in meaningful ways that stick. And then these participants can become your next donors, completing that cycle from engagement to support. So despite the fact that only a few people raise their hands, I'm sure everybody here would like more money to put transmitters out or get more bands or be able to open up a new site. So I'm happy to talk to you about how we might be able to do that and help you do that. And with that, I will answer any questions.
New York City Audubon is an organization that works for the protection of wild birds and habitat and their habitat in the 5 boroughs of New York City. So improve the quality of the life of all New Yorkers, birds and people. We work in 3 main areas: conservation, engagement, and advocacy. We have several projects in conservation, and with the information we collect from these conservation projects, we use that information to engage the people, the people of New York City. With that information, we can let the people know how many birds we have, what kind of birds, and what are the problems that the birds have in the city. We also do advocacy on the birds. We're helping passing several laws. For example, the bird collisions. So now there is a new law in New York City that all the new construction and new retrofits need to use bird-friendly glass in their construction. We're also working in another piece of legislation that is about the lights out in New York City, to turn off the lights during the migration of the birds. So we have several projects. But today I'm going to talk to you. Oops, sorry. I don't know. Nothing. I'm going to talk to you about conservation, what we do in conservation. We have 3 projects, 3 main projects. We have several others, but I'm going to talk about these 3 projects for now. We have a project in green infrastructure. That is a very important part in New York City. It's a concrete jungle there, so we need as many green spaces as possible. So the green roofs they're being increasing in New York City. So that's a very important part for the wildlife to use. So we have a project that goes with the green infrastructure. We also have a project that we call a project Safe Flight, that is about bird collisions on buildings. You can imagine New York City we have a big problem. We have hundreds of birds colliding with buildings in New York City. But we also have the bay there. So we also work with waterbirds on the New York Harbor. And that's what I'm going to talk about mostly this time. We've been banning all kinds of birds for a long time. We started in 2006. We've been banning 83 species so far, and more than 5,000 individual birds. And we also been banning waterbirds and shorebirds in particular, and waterbirds are the key focus for conservation. So these are the waterbirds that we've been banning. And today I'm going to talk to you about 3 species in particular: the American oyster catcher, the herring gull, and the semi-palmated sandpiper. Okay, I'm going to start with the oyster catchers. We have the largest population of these species in the States, in New York City. And we work in this area that you can see here. This is Manhattan, and this is a place called the Rockaway Peninsula. We work at the end of the Rockaway Peninsula in a place called Breezy Point. We have 4 sites where we monitor productivity of the population in general. We monitor also individuals and their movements. We've been tracking this population, population of oyster for over 12 years. And we've been banding adults and also chicks. So far we've been banding 154 individuals, both adults and chicks. And the information that we collect with the banded birds is very important, and we get a lot of information from that. As an example, I'm going to show you this graph. These are banded individuals or banded pairs that we've been following over the years. So, for example, you can see this bird we've been following for 10 years. We know that it's been nesting in the area for 10 years. And we can get a lot of other information. For example, this guy, the black is the color of the band, and this is the code. This bird was banned in Virginia in 2007. So this bird is 17 years old, and it's been nesting in our area for at least 9 years, probably more, but we don't know that. Other information that we collect with this is the reproductive success of the fledgling success of individuals or pairs that we are banded. For example, I'm going to talk about this guy again, the black JC. You can see every line is a year, and that is the number of fledgling pairs per year. So that guy on top has been very successful over the years. There have been several years that it fledged 3 birds, or other years that it fledged 2 or 1. But you can see all the birds that we are able to follow because they're banded, because we banded them. And you can see the reproductive success vary a lot. But it's a very important information, very detailed information about the population that we have there. Another information that is also very important for us is the movements of the oyster catcher when they leave the area. So we have a site of oyster catcher wintering as far as Honduras. For example, this chick, we banded it last year in 2023, and later that same year, it was reside all the way here in Honduras, in the Pacific coast of Honduras. So we can follow their movements through reside. And also you can see in the map the numbers of resides per state in the Atlantic coast. As you can see, we have a vast number of resides of the birds that we banded in our colony. So that is also very important information for our projects. Okay, the conservation impact of this work is that we can, with all the information we get, we can protect the colony, and we track the productivity, and that helps guide the management, the people that manage the area about the best way to protect these species. We also work with the American Oyster Catcher Working Group. So we have a vast network of researchers along all the Atlantic coast of the US. So we have very, very good information about the whole population in the US of these species in the Atlantic. Also, the colony in Breezy Point help us engage with the public. We can show the public the birds there, how they're doing, what they're doing, how important it is to protect them. Okay, and now I'm going to talk about the semi-palmated sandpipers. We've been banding semi-palmated sandpipers since 2012, if I remember correctly. And we use the sandpiper as an umbrella species for all the shorebirds that use New York City to stop there. So we can follow their movements. We band the birds, and we also use nanotags, and we use the Motus network to follow their movements. So we capture this bird in this area, Jamaica Bay. Most of you might be familiar. This is New York City, this is Jamaica Bay. And this is a very important stopover for shorebirds in general, because they stop there for several days, they replenish, and they continue their movement, either north to their breeding grounds or in the way back south from the breeding ground. We can use all these data that collect with the movements of birds. The map that you see there is all the birds that we've been putting nanotags on, so you can see all the movements of the birds there. And as some of you know already, there is plan for the installation of offshore wind farms. And as you can see, the area where they planned to put the offshore wind farm is right in the center of the migration route of the shorebirds. So we're hoping that the data we collect with this information will help to understand the possible interaction between the offshore wind farms and the shorebirds during their migration. So the conservation impact of this project is that banding and following the movements of the semi-permanent sandpipers helps to protect not just this species, but all the shorebirds that stop in New York City. And we can use the data that we collect with the oyster, the semi-permanent sandpipers, to see the potential interaction with the wind farms, the offshore wind farms. Okay, finally, I'm going to talk about the herring gulls. You can see this is a nest of herring gull, and it's on top of a green roof in Midtown Manhattan. That you can see all the buildings next there. So the birds nest in that roof. So the gulls are species in decline in New York City because of lack of loss of habitat. So the green roofs are helping replace those habitats that were lost, and the birds are using these green roofs. So the technology behind the green roof is kind of complicated, and I'm not going to talk about it, but it's used to put several layers and to include soil and vegetation on top of the buildings. So the green roof provide habitat for some of these nesting birds, and that helps us work to protect the largest green roof colony of gulls in New York City through science and engagement with the public. The conservation value of this project goes beyond the borders. The work we do in Javits with the gulls is being featured in this program called Extraordinary Birder that you can see in Disney Plus for the New York National Geographic Wild. And this project this information and this colony beyond the borders of New York City. And I was talking with somebody about also the importance of banding the gulls. We band the chicks and few adults in that roof, so we can follow their movements. And as you can see, some of the birds that were born in that roof now are moving as far north as the Great Lakes in Canada or as far as Texas. Sorry, I moved there. As far as Texas. So they are all over the place. And not just that, but also, oops, I don't know what I did, I'm sorry. That computer doesn't operate.
It's just a. All right. Oh,
I'm not don't worry. I can work like this.
it's not showing. There we go. We got it.
Okay, so I was telling you about the birds we banned there. Most of them are chicks when we ban them. And now we're finding that some of those are returning to the tag. These gold, maybe you can notice the orange one here, is a 472, was hatched in that roof in 2018 and returned to build its own nest in 2022. So this is very important information. We now getting more information about site fidelity and other aspects of the biology of this bird. So the conservation impact is that we're established a good relationship with the owners of the building. Before, for them, this species was a nuisance species. They didn't want it there. But through our work with them and our cooperation, they finally realized the importance of have a colony of gulls in their roof. So now we have long-term support from them, and we know this colony is going to stay there for a long time. Also, these birds are very visible right in the middle of Manhattan. So all the people can see the birds and also raise awareness of their presence, not just of the gull, but in wild birds in general. Okay, we're planning to continue with all these three projects. We want to continue with the work with American ostrich-catcher. We're going to continue banding the birds. And we're going to start working in climate change and predation. We just started a project a couple of years ago about camera traps near the nest of these birds. So we know the predator that attack this bird and the dynamics, and we also can get a lot of information with those camera traps. With the semi-permanent sandpipers, we're gonna continue banding and putting nanotags on those birds, and we're gonna keep working to see how they interact with the offshore wind farms, if there is any potential conflict of interest or not. And we're gonna keep working on the green roof. We're gonna ban gold. We're gonna continue banning gold, but we also we're starting a project and we want to ban passerines in the green roof. As you know, some of you know, New York City is a very important migration path for not just for shorebirds, but also for all the land birds that migrate there during the fall and the spring. And they're using the green roofs. So we want to know how they use it and if they come back the next cycle or whenever. So this is an important part of our work. And that's all for now. Thank you.
Next, we have Annie Lindsay. I'd like to welcome her. Annie Lindsay is the bird banding program manager of Powder Mill Nature Reserve in Rector, PA, Field Station Carnegie Museum of Natural History. And we'll be talking to us about refining methods to add stable isotope analysis of migratory bird fat to the ornithological toolbox. Thanks. Thanks.
All right. Yeah, so this is getting a little bit away from using telemetry to track birds, and we're going back to the using isotopes to track birds. All right, let me see if I can make this work. Nice. All right. Yeah, you did it. All right, so we know that birds need to fuel for migration. So before they take off on a migratory flight, they need to increase their food intake, and they put on a lot of fat. This is something that bird banders look for. We score bird fat. So at Powder Mill, we do a scale of 0 to 7. Not that that's terribly relevant, but I'll talk about fat scores a little bit later. We also know that there are birds that can make really long flights, and there are birds that make much shorter flights, so smaller hops. And the question is, how long can birds migrate before they have to make a refueling stop? So there are a few papers out there. So there's some evidence that gray-cheeked thrushes can go all the way from Central America to the Great Lakes, and that's a huge flight. We also know that there are birds that can do the trans-Gulf, I'm talking about spring migration, the trans-Gulf flight and arrive on the United States Gulf Coast with plenty of fat that they could potentially continue farther north. But there are other species that maybe take shorter flights, these smaller hops between habitat patches. Okay. I'll also talk a little bit about stable isotope analysis. I don't know if anybody's familiar with this. This is something where you can take a sample of a tissue from an animal and you can look at the stable isotope signatures, and that tells you something about where that tissue was grown. So rain specifically has a deuterium signature that is incorporated into producers, and then when consumers eat that, those
those stable isotope values they move up the food chain. And so this has been studied in different tissues, so primarily blood, claws, feathers, things like that. And there are other stable isotopes, right? Carbon will tell you something about the habitat, same with nitrogen, and there are several other things that I'm less familiar with. So anyway, it rains, plants grow, caterpillars eat the plants, birds eat the caterpillars, and then we take a feather sample, we can learn something about either the geographic location if we look at hydrogen isotopes, or habitat if we look at carbon and maybe nitrogen isotopes. Okay. And we've been able to look at that offset between the isotopes in the feathers or whatever other tissue and the local rainfall. And they've created these isoscapes. So this is for feathers. This is what deuterium looks like in feathers. You can kind of locate, maybe not pinpoint, but you can locate on a map where a feather or blood or claws were grown. I should also mention that there are some tissues that are inert. So once the tissue is grown, that is the value that you're going to get out of the isotopes. And there's other tissues that there's a turnover that you have to take into account. So blood is continually turning over, right? It's not, once it's grown, it's always turning over. So my question was, can we look at bird fat, right? Because this is something that birds are on stopover locations. They eat a lot, they grow the fat. Fat is relatively inert once it's grown. So if we catch a bird after it has arrived to a new stopover spot, can we take a sample of the fat, the residual fat, and figure out where that bird was before it arrived at our banding stations? And this has not been done yet. So here's my study site, Powder Mill Nature Reserve. It's in southwestern Pennsylvania. We're located between two ridges. It's really great habitat, just nice forest habitat. So birds do seem to stop over, but they seem to also filter through our area rather than on the Gulf Coast. That's a geographic barrier, so things may stop before they head south, or once they're heading north, they see that first land and they fall out. So we are a slightly different type of stopover spot. We operate a year-round banding station. We started in 1961. We have over 800,000 banding records now. And so this is an aerial photograph from a couple of years ago. This is just what our banding site looks like. So it's transitional habitat. There's some field, there's some surrounded by forests, and there are these really nice ponds. So that's a lot of things for birds to eat as they're migrating, and in the winter and the summer too. Okay, so I have 3 questions that I wanted to ask. So because this really hasn't been done, especially with wild birds, I wanted to know if sample preparation would affect the percent yield. So we want to know how much lipid we can get out of a fat sample. So if you sample fat, there's going to be other things in it. There may be connective tissue or blood or whatever. So we have to purify the lipids first. So I wanted to know, does the sample preparation method affect that? And does it also affect the stable hydrogen and carbon isotope values? So my second question is it a repeatable measure? So if I take a sample and divide it into several small samples, is that repeatable? Am I going to get the same results? And then the third question, and this is the big one, can we actually use the stable isotope values in the lipid samples to assign a geographic location to where that tissue was grown? So in this case, fat tissue. So I started using chicken fat, so just a chicken breast that I got at the grocery store. So I don't know where that chicken came from, but I knew that that was one sample of fat. It's one bird. The fat should be consistent within that one chicken breast. And so what I found is that extraction method does not affect yield. So you can just get about half of the sample, you can pull about half of the sample can be lipids. You can extract about 50%, give or take. And I think that most of the variation you see there is because there's some residue that'll be left in the tubes, and maybe you spill a little bit on the lab bench. But you really get, so if you take 1 milligram of fat, you can get 0.5 milligram of lipids out of it. So also using chicken fat, I wanted to see if, based on what purification method, did that affect this isotope values? So there are, in the literature, it's a little bit inconsistent how people do things. So some people will freeze dry samples before they attempt a lipid extraction or lipid purification. Some people don't even attempt the lipid purification, they just use whole fat samples. So what we see here, and I feel like it might be a little bit small on the screen, but the letter A, that is, those are samples that were only freeze dried. So they're, I was using binoculars, sorry for the small fonts. I know. All right, so sample A, or it's several samples in group A. I freeze dried those only. So I did not do any kind of lipid purification. Those were whole samples only. And those are the only ones that showed a significant difference between their isotope signatures. So the hydrogen isotopes, it was a little bit higher of a signature. And the carbon isotopes were a little bit lower. So those are all negative values, per mil values. So the other B, C, D, and E, those were just variations of this lipid purification. So whether I soaked it for 48 hours or 24 hours, did I vortex them at the midway point just to sort of mix up the solution, and would that, get us more lipids out of the sample? Letter B was not freeze dried. So I thought that there would be a difference, right? If you freeze dry something, you're taking some water out. So I thought that would affect the hydrogen values, but it did not, turns out. But if you don't purify a sample, yes, those other parts, right? So the connective tissue or blood, that is going to affect the sample. All right, so this is what it looks like. It's these little wads of fat. These are actually wild bird fat. This is not the chicken. The chicken were yellower. I mean, something to eat. And this is what the sample looks like before anything's done. You put them in these glass tubes. You use a chloroform methanol solution, kind of smush it up a little bit to get the surface area of the fat sample, and it comes out into solution. Then you let it evaporate. It usually only takes about a day to evaporate. You need a fume hood. Don't breathe in the chloroform. And this is what the lipids, the purified lipids, look like. It's kind of like a goo, like a viscous goo. Yeah. And this was a little bit of a departure from my expertise, is actually being in a chemical laboratory and doing this. So it was really good practice to be back in doing that. All right, so moving away from the chicken fat, I wanted to know if this was repeatable, right? So if I took samples from lots of different birds, could I be assured that I was getting good, meaningful results? So with wild birds, I did, I don't recommend doing your field work during a pandemic when you don't have help. You only have a child to help you because you live in the same house and can breathe the same air. But these were my study seasons, was 2020 and 2021. So in the spring seasons, I was sampling fat from birds that I assumed to have just arrived in the morning. So it was after a nice night of migration based on all of those birdcast, weather migration, all of those maps. These were birds that arrived to powder mill with low fat reserves. So I took fat of maybe 1 or 2. I pulled some fat out and assumed that they had come from somewhere south of us. In fall of 2020, I sampled some other birds that were, I assumed that they were local origin. So what that was birds that we banded with a 0 fat, no fat, and then upon subsequent recapture, they had gained fat. So several days later, the birds had fat. Probably they put that fat on locally. And also in fall of 2020, I did a captive experiment. So this is something that I guess as banders, we have to talk about permitting, right? This is not something that is, none of this is covered under our BBL or BBO permits. This is Fish and Wildlife Service permits. So I held Swainson's thrushes in captivity until they gained fat. So they had constant water and diet. So we knew it was controlled. They were in an, I'll get there, in an aviary, but I'll get there. All right, so I don't have pictures of actually doing the fat sampling because I couldn't do it and hold a camera at the same time. So there's this paper, this Rocha et al. 2016, where they talked about this method of being able to take fat out of birds without sacrificing the birds. So that was something that's important to me, is to not harm the birds. I don't want to sacrifice them. I'm not saying it's bad, but that's not what I wanted to do. I wanted to see if you could do this and still let the birds go. And this paper indicated that yes, there were not negative effects. If you let them heal, it was fine. So this is what bird fat looks like, but I didn't sample birds that were that fat. In fact, I found that the fatter the birds, the harder it was to get the fat out. It was just really slippery and you couldn't pull it out. So the way that this worked was if a bird was an appropriate candidate, I numbed this, the furcular area, with lidocaine and prilocaine. So it was, I guess, hemorrhoid cream, because that's what came up in my searches when I was trying to find this medicine. So you numb it, you let it sit for a little bit, you make a little incision right here, you use forceps to pull a little bit of fat out, you snip it off, let the rest of it fall back in, and then you can use veterinary adhesive, so that's super glue, to close it up. I put them back in a bag, let them sit, tested for pain response. None of them seemed to show it. They seemed to heal up just fine. Well, they weren't bleeding, I guess. This rarely hit a capillary, so that was really good. And they all flew when I let them go. So that was evidence that things were going okay, at least at the beginning. And I'll get there. No, I have no evidence this is bad, so that's spoiler. All right, so with my captive thrushes, there is it's still there. This is Squirrel Town. So this was a structure that they used to look at thermoregulation of flying squirrels. It hasn't been used in maybe 20 years. So we repurposed it into Thrush Town. And it was great. We put a roof on it to keep water, rain, whatever out, but it was open air. And there were these cages on the inside. We put different cages in, so each thrush had its own cage. Several times a day, we would put fresh water in there with them, and waxworms. And this is my assistant. So I did put vegetation in there with them. So this is one of them. And these were the birds that we waited until they gained fat, and then sample and let them go. So that was between maybe a couple of days and up to, we didn't actually reach the 2-week mark, but that was what the permit said, was up to 2 weeks. There was one bird that did not gain fat, but it was molting. So this molt migrant. It was a little bit early. It was molting. I was like, all right, you're not going to gain fat. I'm just going to let you go so you can molt in peace. But otherwise, so this is what it looks like. Based on these captive birds, it's very repeatable. So what I have here is, this is all of the captive birds. So it's about 10 birds. And if you look at the standard deviation at 2.6 per mil, that's in line or better than a lot of isotope studies. And the other two, these are individuals where I had a big enough fat sample that I could run triplicates. So that's very tight also. Excuse me. All right, so now on to more wild birds, but not captive birds. So the question is, can we actually use this to determine where birds were before they arrived at our site? All right, so these are the birds that were captured during spring migration. So 2020 is the blue dots, and 2021 is the yellow dots. So the sample size is a little bit small in some of these species, but it's showing us a general trend. Another thing that we have to think about is, I don't think that every single individual of a species is going to spend the same, be in the same previous stopover site. So there's going to be some variation there. But the trends that I saw were that magnolia warblers are the only species of these ones that were sampled both years that showed a significantly different hydrogen value between the years. So that, there are probably year effects here. So what did the weather look like? Were there nice, strong tailwinds? Were they making bigger flights or shorter flights? Different years. Swainson's thrush and hermit thrush was the tiny sample size. There were no significant differences between the years in the hydrogen isotope values. Another thing that was very cool was that Magnolia warbler and Swainson's thrush, the two that had the highest sample sizes, did show some differences. So in reading this graph, the more negative the number is, the higher the latitude, so the further north the bird is. So it looks like Magnolia warblers were closer to Powdermill at their previous stopover site than Swainson's thrushes. So that kind of tracks, right? If we think about thrushes and those gray cheek thrushes that can make those huge leaps, I don't know if Magnolia warblers can or not. They have shorter wings, they're smaller birds. But all three thrushes look like they were somewhere much further south than Powdermill than Magnolia warbler and catbird. So I think there is something to this. I think it needs to be refined quite a bit because during fall migration, these were the presumed local origin birds and their signatures are completely different, right? Like you would expect these to be lined up. But there might be other things to this. The red-eyed vireos, they do breed at our site. So I feel pretty confident that those are showing us local hydrogen isotope values. Northern water thrush, they don't breed there. They breed further north, but they stick around at Powdermill for up to 2 weeks or more. Honestly, they're a really long stopover species. So I don't really know why they're so low. But again, the sample sizes are pretty low. So this is a technique that I think is worth investigating. One of the avenues that I think would be worth investigating is when there are collision birds that don't make it and they're going to a museum or institution to be added to a collection, take fat out too. And freeze it. Negative 80-degree freezers is how we store them. So yeah, if you have that. All right, so is it safe? And there's evidence from other studies of this sort of somewhat invasive procedure in the literature that says, yes, it seems safe. But a lot of that literature is in shorebirds or bigger water birds, like, well, killdeer is one I can think of. And then there's some other like, not albatross, but some just bigger birds. And so I did this analysis that's, it's supposed to show, right, like these are really low numbers compared to just non-biopsied birds. So we're trying to compare really low sample sizes to bigger sample sizes. And it's just showing, is the recapture rate for the biopsied birds any different than for non-biopsied birds? And all of those p-values being way greater than 0.5 or 0.05 shows us that yes, it's safe. There was no difference in the recapture rate. There is major difference in the recapture rate between species, and that's why I separated them out by species, because I didn't think you could compare a Swainson's thrush that we rarely recapture to a catbird that we recapture 50 times a season, right? So anyway, yes, at least in the short term, this is a safe procedure. We don't have data for long-term is it able to migrate and come back? Does it affect it? It seems like, so based on some other literature, when we talk about removing fat, are we removing a bird's ability to get fat? It seems like they can regenerate fat cells. So it's going to take some energy from them. They may have to pause and refuel for longer, but it seems okay. And when we recaptured them, we did look at the incision and that they were healing. So they would scab and then sort of knit together and it looked fine. So yes, it's safe. All right, so the question is, can we use the stable isotope analysis to try to figure out what a bird's previous stopover location is? And I think that this is a method that has potential, but it needs a lot more work. So had COVID not happened, the plan was to go to other locations, other banding sites and very different latitudes and get fat samples from those birds. Didn't happen, but that could be a really good thing. This is why a nice network of banding stations and collaborations is important. So once you have a lot of samples and you come up with these calibration functions, and you can make the isoscape, just like in that previous slide, but for fat. All right, does anybody have any questions about that?